Hi everyone, thank you very much for joining this session today. I'm Joel Roca and I'm a postdoc researcher in Christine Norengo's lab at UCL in London. And today we'll talk about the work we've done to identify novel PETases, that are enzymes that have the capability to degrade PET, it's a type of plastic, using a computational classification system. So first of all, I think we're all probably aware of the plastic problematic and how it has increased during the last decades, the production and therefore the accumulation of these types of material. There are many different types of plastics. The most common one, for example, is polypropylene. But in this particular research, we're interested in PET for a number of reasons. First of all is that it has a very low decomposition rate, which means that, for example, for a single bottle, it would take around 450 years to degrade by itself. They are very often single-use products, which means that they get accumulated very quickly. And the current recycling methods that are available are very inefficient and drive to the loss of properties of the material, which doesn't make it 100% recyclable nowadays. Plus, it uses normally mechanical or thermal processes, which has a very high energy cost. But actually, the real reason we are quite interested in our group on working with this target is because almost 20 years ago, it was identified the first enzyme that could promiscuously degrade PET. And this enzymatic biodegradation is a very promising way of recycling PET because it generates a set of monomers like BHET, that is bihydroxyethyl ethylene terephthalate, that are very easy then to recycle and then to re-include in the circle of producing new plastics using exactly the ones that were disposed. So the first one identified was an enzyme, a cutinase from Thermobifida fusca, because those enzymes are not normally, as I mentioned, promiscuous. Their normal target is not PET. It's just that they have the ability of degrade PET because their natural substrate is quite similar. A few years later, in 2012, another enzyme with a very high activity also in this case was identified on a set using a metagenomic sample from leaf branch compost. This cutinase is still nowadays one of the most active ones, but also with the other cutinase identified in 2005, they are both thermophilic proteins, which means that they need a high temperature to be active and to have a good degrading activity, which also implies that if they were to use industrially to degrade plastic, the energy cost would also be very high. But luckily, and quite interestingly, in 2016, another enzyme was identified with the ability of degrade PET, but in this case, it was a mesophile, which means that it can act almost at room temperature. Actually, I think the best temperature for its degradation is around 35 degrees, and this is ISPETase. It was identified on the bacteria Dionella sakaiensis in a plastic recycling factory in Japan. And it's quite interesting because it also was using PET in that factory as the sole source of carbon. In 2017, since that discovery of ISPETase and the years after it, we've been having more and more variants of this enzyme that have been improving the activity of ISPETase, like thermopestase, diaspetase. I don't want to go into the detail, but the most important thing here is that most of those have a quite similar sequence to the identified ISPETase. If we take a look at the PETases database, that is this database that collects all the information available on enzymes that with the ability of degrade plastic, we can see that most of them have a sequence identity around 40 to 50%, which means that considering these enzymes belong to the alpha-beta hydrolase superfamily, that is a massive superfamily, the sequence space that have been sampled so far is very small. This is why we came up with the idea of trying to identify more of these enzymes farther away in sequence from the ones that are already known using metagenomic databases like Magnify. And for that, we're using a computational classification system to try to fish the ones that we think could be the most new active PETases. So this protocol starts by filtering, as I mentioned, this massive, overwhelmingly big magnified database that contains at the time that we did the search, contained already over 1 billion sequences. Now it's even bigger. And the first thing we did was extract all the proteins that belong to that alpha-beta hydrolase superfamily. That was still over 500,000 different protein sequences. Then, to stay a bit close to the known PETases we already had in literature, we performed 2 rounds of jackhammer search to define what we called our PETase zone that contained over 20,000 different proteins. Then, this PETase zone, what we're doing is classifying into different functional families using our in-house protocol, GemmaFunFamer, that essentially is based on two different steps. First of all, it builds hierarchical trees based on sequence alignments and profile comparisons, but those alignments are further split based on the sequence conservation that we have, on the differentially conserved positions that we have across those functional families. Because if a position is conserved to a different residue in different functional families, that means that it's probably quite related to the function or the specificity that that protein has. And actually, just to mention, this protocol has now been updated. It's currently under review, but we hope it will be out soon. And in this case, it uses embeddings instead of just profile comparisons, which makes it more sensitive and, even more importantly, much more faster, allowing us to handle many more sequences at once and much bigger superfamilies. So using this protocol, we classified those over 20,000 proteins into almost 1,800 different functional families. But then we further filtered them down to keep only the ones that have a high sequence diversity, because then we know that the positions that are conserved are significantly conserved for those functional families. And moreover, we also restricted to the ones that were quite close to the functional family that contained that already well-described and characterized, and it's a good active ISPETase. With this, it's a quite stringent filter. We ended up with 84 FAMFAMs that we analyzed in detail. If we built a tree of those, a hierarchical tree for those 84 different functional families, this tree was based on the HMM distances built for each of those FAMFAMs. This is what allows us to perform our next step, that is this tunability analysis. With this, what we want to do is identify the positions that across these 84 functional families are differentially conserved and assign a score that allows us to say which positions tend to be more tunable. And with this, I mean that those could be positions that are more interesting to mutate or more interesting to study because they are very likely to be involved in the specific function or specificity of that protein. Just to give you here an example, what we're using for this tunability computation is just the Grantham score that computes the chemical distance between different residues. If you check, for example, the position highlighted in yellow, we could have that in the functional family 1, we have a phenylalanine, and in the 2, a tyrosine, which are quite similar residues with an aromatic side chain. This would have a very low tunability score because we don't see that position is changing a lot from functional family to functional family. Otherwise, on the other hand, if you take a look at the position highlighted in red, we can see that sometimes it may have an aromatic residue or a polar residue or an apolar residue, which means that position is conserved to very different amino acid types, which makes it quite relevant in terms of how it could be involved on the actual function of the protein. And this is what we call these positions with a high tunability score, function determining positions. With this, we identified 22 different positions that are around the binding pocket of this enzyme. And if you take a look, this structure is ISPETase, and you can see here we're a docked monomer of PET. You can see that the positions that are more tunable, the ones that are highlighted that are shown in red, are the ones that are closer to this monomer in the structure, while positions that are a bit more farther away from the binding pocket have a lower tunability score. Based on this analysis and considering the fact that originally we just randomly picked few sequences across the tree, but none of them were actives, we restricted ourselves to three functional families, that were quite close, or even including the one that contains ISPETase. So we know that we stay close to that one, and then therefore it's more likely to find actives, despite we're restricting ourselves a bit in the sequence space we're exploring at the moment. If we zoom in and we just take a look at the sequences contained within those three functional families, we can see that they cluster quite well together. Functional family 32, it's here on the right mostly, and the functional family 143 that contains ISPETase, and 26, you have it here on the left. And but these were still too many sequences. These are around 300 different sequences containing both magnify entries and also sequences from UniProt. So we developed a score to decide to rank them and check and to select a small set that we could experimentally test with the help of our collaborators. So that score is based first on three different components. We include these SoluProt predictions because the first, in some of the rounds we had at the beginning, we had many expressibility and solubility problems on the lab. So our experimental collaborators were very, of course, they are very interested in the fact that they can actually work with the proteins that we suggest. So it's an important factor to take into account, and actually it's quite one of the trickiest bits to when we come up with new sequences. But then the two components that I will talk a bit more in detail is that we were trying to find sequences that showed a certain distance to the selection we already had. So starting from ISPETases, we wanted to find things that are far away in the sequence space, but that at the same time that showed a certain similarity to ISPETases or the known PETases in literature in general. For the distance, we just take into account again the Grantham score. So across those 22 different positions that we know are differentially conserved across these FANFAMs and that we think are quite important for the function of this protein, we select the ones that have a higher distance. And if you take a look, for example, at those 10 different selected magnified candidates, you can see that this is the Grantham distance to ISPETase across the 22 functional determining positions we chose. You can see that some of them, the score, it's white. The squares here in the heat map are white, which means that that residue is completely conserved and it doesn't change to the wild type, to ISPETase. But in some other cases, in more or less half of them, we have clear differences, and we're going a bit farther away on the sequence space based on those 22 selected positions. That's for the distance component. And then for similarity, we're essentially taking into account three different parts. First of all, we're checking whether those sequences contain mutations that are already well known and well characterized in literature as a good mutation for PET degradation. We're also docking the monomer to verify that the binding pocket is white enough, for example, to actually degrade PET. And finally, we're checking several biophysical properties of the binding pocket, but I will not go into the detail there. So with this scoring and by picking up those 11 sequences that you have highlighted here, we identified 3 new actives with the help, of course, of the Florian Holfelder's group in Cambridge, that they can work up and that they can derive to, despite they are not very, very good actives, so they don't have a very high catalytic activity, they can work them up and try to evolve them because we're a bit farther away on the sequence space now. For example, one of the actives, that it's magnified 10, has only a 24% sequence identity to the wild-type PETase. Now, if we take a look, this is essentially showing that same tree with around 300 sequences, but showing it in a more visual way in a sequence similarity network. You can see that the actives that we identified we have 2 here that are relatively close to the functional family that contains ISPETase, but we also found one in functional family 32 that is a bit more farther away. And here we also identified in those different blobs, we also identified few more negatives. We were able to pick up sequences from different blobs because we're using these distance components. We're trying to find things that are actually far from this ISPETase cluster here. But if we take a look and we take one step back and go again to those 84 different functional families that I talked at the beginning, the only part we're currently sampling at the moment and that we're addressing is this one here. We're still checking a very small part of the sequence space of the hydrolases, and we're just restricted to those 3 functional families at the moment. So now, and to talk to you a bit about the next steps we're taking, we want to find ways of which functional families could still be interesting here, because there are many that, of course, may not have PET degrading activity, but we really believe that they may still be some out there that are quite interesting, but that we are not able to find with the current sequence-based techniques we've been using. So what we're doing now, and thanks to that first round that we were able to identify new actives and also inactives, which is very important for our tools, we built AlphaFold models for the different magnified actives and inactives, and we're starting to find certain patterns in that binding pocket that now we want to use as a prompt to try to sample and try to go around those functional families in a different way than the approach we had before. Moreover, we're combining this with sequence embedding methods that are also providing much more information, a much richer description on the proteins than just using the sequences. Just to finish with a quite alarming fact, only in Canada, around 3 billion kilograms of plastic are produced each year. And from those, this data, I just got it recently from the Government of Canada website. You can take it yourself. From those 3 billion kilograms, only 9% is recycled. This, of course, it doesn't only include the PET, but other plastic polymers. But this means that only during this around 15 minutes presentation, 80,000 kilograms of plastic have been mismanaged. I mean, probably only today on the lunch boxes that we had, that was an important amount, to be honest. And finally, just to acknowledge the different people that have contributed to this work. First of all, the group in CAF, and of course, Christine for her great guidance always, and the different members that have contributed or that are related to this plastic degrading task, like Clemens, that did a lot of work. It's a former postdoc, did a lot of work on that I've been presenting today, Jude, Wayne, and Nico. Also, our experimental collaborators that run all the wet lab processes for PETases, in Florian Holfelder's group, Josephine Marian Angorian, and finally the people at Rob Finn's group in EBI that provide all the Magnify support. And of course, thank you to all of you for your attention. Yeah, I'm happy to take any questions.
Any questions?
Do you have any online questions first before I go? All right, thanks. Great talk. So can you talk a little bit more about your experimental validation pipeline, maybe? How fast is this going, and so on?
Well, we don't run. You might imagine we don't run our.
No, I know you don't run your own.
Ourselves.
You have a collaborator, right? Yeah.
It's always a bit of a fight to get them to test sequences because I mean they need to trust them enough. It doesn't take too long to filter them. They use the current standard techniques for that. That I think it's a small PET disk that they put with their protein solutions that they express, if I'm correct, in E. coli. It doesn't take too long to test. The problem is that then you need to sequence. If you want to test many sequences, they normally only sequence the one that seem more active because they have also this directed evolution platform, which they can do high throughput evolution of those proteins. So it depends. It depends exactly on what you want to get out of those experiments. If you just want to know if a sequence is active or inactive, I think it doesn't take that long, but you cannot do it high throughput.
How many have you managed to look at?
From that we had a first round of around 10 to 15 that we didn't find any active. And then with this more refined protocol, we tested 11 and we found 3 actives. And now we're hoping to send some other batch of 20 soon.
Thank you.
I might have missed this, but what's the ultimate goal of this? Is this to look at plasticity of families in general, or is it to get a more active, like to start some, if they're doing a lot of directed evolution, they could start with the ones they have that work and just beat it to death to get a better one. But this is to kind of start from other places in the evolution space and see if you can get better ones. That's the idea?
Yeah, exactly. Yeah, of course, it would be great if we could able to find the holy grail of PETases and metagenomic databases. But we think that with these promiscuous enzymes, that's not really possible. So what our collaborators are very interested with, it's in going very far away in sequence, because we think that maybe with the current sequences that we know, we're in this sort of like local minima that we cannot really get any better at this point. So we want to explore really some stuff farther away and try to see if we get something that can be upscaled industrially.
Oh, very nice talk. Okay. Very nice talk. You might have mentioned this at the beginning, maybe I missed it, but I'm thinking these enzymes didn't evolve initially to degrade plastic. So I was wondering, do you have any idea what their maybe original substrate might have been, and could that sort of group of substrates also suggest other proteins that might also degrade plastic?
Yep. Actually, we've taken a look at this sort of thing to focus on the natural substrates and how similar they look to PET to try to find more targets that way. For the most common ones are cutinases, that the natural target is cutin. It is this wax that leaves have on top to make them a bit more hydrophobic. But then ISPETase it's a quite interesting case because it's the one that was identified that only uses PET as a source of carbon, at least in that plastic factory. And it means that it has evolved to do that in around 50 years, but no longer than that. But we still think that, of course, there is a lot of room for evolution in this case.
All right.